I've never addressed a large audience of mycophiles before. Knowing that this was the Telluride Mushroom Festival, I will focus a good part of my talk on fungi, but not all. So I tried to pick some things that are samples of things I've done that will be of interest to you. Some of it will be review for some of you, but probably not all. So let's get started. Psychedelics. What is a psychedelic? The scientific definition that I always use is there are substances that produce changes in thought, mood, affect that occur otherwise only during dreaming or at times of religious exaltation. And so I know nobody in here has ever experimented with psilocybin mushrooms, but any of you that maybe have will get that definition pretty easily. So this is the one that's really famous, and it's becoming more famous all the time. It's the mushroom that the Hefter Research Institute decided to focus our clinical studies on. And all the things you read in the paper, in the media these days about psilocybin being used to treat depression and anxiety and addiction, etc., that's all work that we started at the Hefter Institute that I really catalyzed in 1993. These, of course, are teonanacado, flesh of the gods, or food of the gods, hearkening back to this definition of a psychedelic as something that produces thoughts and feelings that occur only during times of religious exaltation or dreaming. Flesh of the gods. They contain this simple molecule, psilocybin, which has a phosphate group attached here, where this R group is a phosphate. And psilocybin is very stable. You can recrystallize it from boiling water. If any of you have made a tea from dried mushrooms, you know it's stable. In the body, this phosphate comes off to generate psilocin, which is a hydrogen there. Psilocin is chemically unstable. If any of you ever had psilocin in a bottle, it starts turning dark pretty quickly. But psilocybin that was made by Sandoz back in the 50s was stable into the 1990s with no degradation. A very stable, interesting molecule. It came to the attention of Western society with this 1957 Life magazine article. Gordon Wasson was a New York banker and amateur mycologist, and he and his wife Valentina were interested in mushrooms. She came from Russia, where they were fungophiles, and Gordon was fungophobe, I guess, and they wondered why there was this difference in cultures. Well, they heard about these mushrooms in South America, so they traveled down there, and this is the Life magazine story. South America is littered with these mushroom effigies. These are the ones that are remaining. Hundreds are remaining after the Spanish missionaries failed to destroy all of them. But they represent, obviously, some sort of a totem. And then Gordon Wasson went to Mexico and had an experience with Maria Sabina. She gave him the mushrooms, and that began our Western knowledge of what these actually were. Although they'd been used for millennia by the indigenous peoples in South America, Western man only became aware of them in 1957. These mushrooms may have a much longer history than most people think. Many of you have heard of Soma. It was written about in the Rig Vedas, the ancient literature, religious literature of India. There were many hymns written in worship of Soma. We drank Soma, we became immortal. We came to the light, we found the gods. A lot of speculation about what Soma was. So in 2009, there was a tomb in southern China that was excavated and contained woolen textiles that had this depiction of a Zoroastrian religious ceremony. This is a priest here over a sacramental fire, and he's holding what appears to be Psilocybe cubensis. Now, there's no reason for a mushroom to be involved in a religious ceremony unless it had some religious import. So this may well have been one of the active components of Soma, which is what it is has been lost to antiquity, but it may have been psilocybin mushrooms or some admixture. In ancient Greece for 2,000 years, there was a ceremony in the village of Eleusis, about 15 miles outside of Athens, and any adult Greek who had not committed murder was privileged to participate in that ceremony. It was called the Eleusinian Mysteries, and they drank a brew called kykeon. This is the bowl it was dipped out of. Presumably there were candles in these outer rings. It was dipped out and given to the adherents. And they had attendants in this. There was a whole temple built, the Telestrion. And there were attendants in there. There was chanting. There was all-night singing. We don't know much about this mystery because if you talked about it, you were killed. This went on for 2,000 years. Any great Greek you could name must have participated in this ceremony. It was like a grand bar mitzvah or something. Here's a scene from the Eleusinian Mysteries, and what's she holding there? A mushroom. Now, I'm sure they weren't talking about making mushroom stew or anything. That mushroom was there for a reason. And the nature of what kykeon was is also a mystery. We know that it contained barley and mint. So there's been all this speculation, well, maybe it was ergotized barley and something. It may have just been a red herring. It may actually have been a mushroom that was involved. So we think the use of these things may go way back into antiquity. We just have lost the information. So there's some other related South American snuffs. Anadenanthera is a South American snuff that's widely used, contains bufotenin, which basically is thought not to be active, but they blew this stuff up their nostrils, so clearly it had some kind of a psychoactive effect. It's a tryptamine, very similar to psilocybin, except this hydroxy group is moved over to this position instead of being up in the 4 position. And DMT, probably everyone in here has heard of DMT. Rick Strassman wrote a book called DMT, the Spirit Molecule, made a movie, DMT, the Spirit Molecule. I actually synthesized the DMT that he used in those studies. And it's in a number of plants. Mimosa hostilis has a lot of it in the roots. You can extract it out. It's a really potent psychedelic. It's active if you insufflate it or you smoke it. Lasts about 15 minutes, extremely powerful, one of the most powerful psychedelics out there. Again, it looks a lot like psilocybin, except it doesn't have this hydroxy here, and it's not orally active, whereas psilocybin is orally active. Most people have probably heard about ayahuasca. Ayahuasca is an admixture of a brew that contains the beat-up vines of Banisteriopsis caapi and Psychotria viridis leaves. Psychotria viridis, or chacruna, contains DMT. DMT is not orally active, but in the Banisteriopsis, which are these red pieces here, which they hammer and shred, when they're boiled together, it extracts out alkaloids called beta-carbolines. And those block the enzyme in the liver that breaks down DMT. So if you take these beta-carboline alkaloids and would just take DMT orally, it would be orally active. So ayahuasca is essentially an orally active DMT. And I've spoken with people who have taken ayahuasca and also used psilocybin, and they say they're basically fairly similar. But of course, there's a lot of interest in ayahuasca because people go to South America and take it and report all kinds of healing miracles. Then we have the toad lickers. Anybody heard of toad licking? So BufoAlvarius is a toad, and in these parotid glands, if you squeeze those glands next to a piece of glass, it'll squirt out this milky white toxin. If you ingest it, it's toxic and you get really sick. But if you let it dry and scrape it off with a razor blade and smoke it, the toxins are peptides and other types of things that don't volatilize. And 5-methoxy-DMT is in there. And you see it looks a lot like bufotenin, except it has this methoxy instead of hydroxy. That may be one of the most potent psychedelics out there. And if you've ever talked to anybody that smoked this toad venom or took synthetic 5-methoxy-DMT, it will really knock you for a loop. It's short-acting, 15 or 20 minutes, and people report profound experiences after smoking this. It was also used as a snuff by South Americans. And here you see this was taken from the inside roots of arula, and they powder it, and it's a red powder, and they blow it up in their nose through a hollow tube, and it's very potent, and it's also related to the tryptamines. It is a tryptamine. Now, chemists have exploited those by doing a lot of different things to see. I did a lot of this work, other people have done this work to see if we could improve on the properties or make them more useful or just to understand how they work. And essentially, you can attach things into the side chain. You can modify things that are on the ring here. And I'm not going to spend a lot of time on that because it would be chemistry that would probably bore most of you. But you can do that and make a lot of analogs. And some of the things you can do, for example, whereas tryptamine itself is not orally active, if you add a methyl group here at this position, the CH3, you get an active compound. And that's because it blocks the degradation of the side chain by the monoamine oxidase in the liver. 5-methoxy-tryptamine itself is also not active, but if you put an alpha-methyl here, CH3, it becomes orally active because it blocks the breakdown by monoamine oxidase in the liver. And just some things that have been done. If you extend those alkyl groups, so here's DMT, it's not active orally. 5-methoxy DMT is not active orally. And psilocin is active orally, and I'll talk about why that is. And if you put large alkyl groups on here, like isopropyl methyl, you can convert it into an active compound, or diisopropyl, isopropyl methyl, diisopropyl, diisopropyl, diisopropyl. Put bulky things around the nitrogen, then they become orally active because it keeps the enzyme from breaking down the side chain. So why is psilocin orally active, whereas DMT is not, and bufotenin is not? So not orally active, not orally active, orally active. And what I've shown is some things here. This pKa is a measure of how basic this nitrogen is. So these are organic bases. In everyday life, something like lye would be a base, a strong base. So alkaloids are bases, they're bitter, and they have a basic character. The pK of bufotenin is 9.87 or 67. That's a pretty basic nitrogen. If you look at psilocin, the basicity is dropped down, and that's a log scale, so that's about an order of magnitude, 8.47. So it's less basic. The nitrogen is less basic. This log P of the partition coefficient is a measure of how lipid soluble the molecule is. So bufotenin, the log P is 1.19, but the log P for psilocin is 1.45. So it's more lipid soluble. So moving that hydroxy from the 4 position to the 5 position has a dramatic effect. It changes the basicity of the nitrogen and also changes its solubility in lipid or fat. So back in 1980, I asked the question, why is that? We postulated that what might happen is a hydrogen bond might form between this OH and the nitrogen in the side chain. And normally you don't see that in a big ring like that. You see it in smaller rings. But we actually did nuclear magnetic resonance studies and showed that there was a stabilization, that this hydrogen bond was very stable. This is a model of a molecule that we modeled with the computer. And if you model it, this pulls right around and interacts with this OH. So here's the oxygen, here's the hydrogen, and you form a hydrogen bond with this nitrogen. So what is the consequence of that? So basicity is a measure of how readily a nitrogen atom will take a proton out of solution, a hydrogen ion out of solution. If the nitrogen is already being hydrogen bonded by that OH group, it doesn't have any really desire to pull a proton out of solution. So that OH replaces the proton that would normally protonate it. So that changes its basicity. The other thing that happens is when these things are absorbed, only an unionized molecule is absorbed across membranes to get into the brain. So if you take a molecule that's charged, it has water molecules arranged around the nitrogen. It has to lose those in order to be absorbed into the brain. But if we form this hydrogen bond interaction here, it prevents that from happening, so it makes it more lipid soluble. So psilocin is orally active because of the interaction between this hydroxy in the 4 position and the nitrogen here. That can't happen if the hydroxy is over here in the 5 position. So we know why psilocin is orally active, but DMT and bufotenin aren't. And the other thing that happens when a tryptamine is deaminated in the liver by monoamine oxidase, one of the things that happens is the enzyme, monoamine oxidase, pulls an electron off of the nitrogen. And that's the first step. And I don't know if you can see that, but say this is a tryptamine. This nitrogen, even with the dimethyl, a nitrogen has a pair of electrons, and that makes it basic. One of these electrons has to be pulled off by this cofactor to generate this radical cation. It's an electron with a positive charge. That's the first step in the deamination. There's another mechanism that involves an electrophilic attack, and the same thing has to happen. It has to pull an electron off of there. Well, when this is hydrogen bonded to that 4-hydroxy, it prevents that electron from being pulled off. So this prevents the degradation by monoamine oxidase. So there are 3 different factors involved in making psilocin active. Okay, so I'm not going to spend much time on phenethylamines, Maybe many of you are familiar with Peyote Cactus, Lophophora williamsii. We know it was used as early as 3700 BC. There's been caves found with dried up peyote buttons from that era. It has mescaline, 3,4,5 trimethoxy phenethylamine, very different from a tryptamine. And we've done a lot of work there. We've added things to the side chain. Sasha Shulgin and I did most of this work. We played around with these, moved these methoxys around, replaced this methoxy, replaced these, and you get some really active compounds. Mescaline is not very potent, 250 to 400 milligrams orally. You move one of these methoxys, it's inactive. Move it to a different position, it's inactive. But if you replace that methoxy with something like a methyl, that's active. Replace it with a bromine, you get a compound called 2CB, that's active. And that's related to those changes preventing the side chain from being broken down by amine oxidases. So when a phenethylamine binds, we did a lot of studies to show that they bind to the receptor when the side chain is something like this. And I'm not going to go into those experiments. I'm going to move on to something that's a little more interesting. So this is another fungus, the ergot fungus, Claviceps purpurea. It infects cereal grains, rye, barley, wheat. And these black things are called Sclerotia. This is the inactive form or the hibernating form of the fungus. And these things produce alkaloids that are very toxic. The alkaloids that they produce look like this, where you have large peptides up here, complex peptides. And those compounds, ergot alkaloids, are extremely toxic. It is a fungus. This is one of those ergot Sclerotia that's been put in under conditions where it will sprout. And you can see it is a fungus. It does produce little fruiting bodies. Here are ergot on rye, on tall fescue, on Dallas grass. And fields that become contaminated with ergot are really toxic to grazing animals. In the Middle Ages, we had episodes of ergotism, or St. Anthony's fire. This is a gangrene of the extremities and nervous system effects. More than 40,000 people died from an epidemic of ergotism during the Middle Ages. What these ergot alkaloids do is they cause the blood vessels in your fingers and toes to contract or constrict, so there's no blood flow. And it's prolonged constriction. People were making bread that was contaminated with ergot, and so they would keep eating it. So this vasoconstriction, these constricted blood vessels, would maintain for a long time until they actually caused necrosis and death, gangrene. So this is what gangrene of the fingers and toes looks like. And it produced also a burning sensation. And if you see these, it looks almost like your hand or feet have been in a fire. So it was St. Anthony's fire. And St. Anthony was the patron saint of people who suffered from ergotism. These ergot alkaloids also occur in species of morning glory. Turbina corymbosa was used by the Aztecs when they couldn't get mushrooms. You grind up these seeds and make an extract, and they produce a psychoactive effect. They contain lysergic acid amide, just a simple NH2, and they are active but not as active as LSD. And these are thought to be produced by an ergot infestation of the plant, some sort of a symbiotic relationship between the ergot and the seeds in these plants. The plants don't actually produce the ergot alkaloids. So whenever I talk about LSD, I like to use this slide. I'm not going to ask for hands to see. Is there anybody that hasn't heard of LSD? This is LSD. It's lysergic acid diethyl amide. So this is an amide linkage. These are ethyl groups. It has two ethyl groups attached here. And this ring is an ergoline. And if this was a COOH, it would be lysergic acid. So it's lysergic acid, NN-diethylamide. Its effects were accidentally discovered in 1943 by the Sandoz chemist Albert Hofmann, who many of you have heard of. And if you had a child in the 1940s and earlier that suffered from schizophrenia or mental illness, you were blamed. It was the parents' fault. They hadn't nurtured the child properly. And you had a term called refrigerator mothers. These were mothers that were cold and distant and had not nurtured their children. That's why the children were sick. There was no recognition that there was anything going on in the brains. It was a nature-nurture. It was a nurture kind of failure on the part of the parents. In 1953, serotonin was identified in the brain. Serotonin, 85% of the serotonin in your body is actually in your intestines, in enterochromaffin cells. It's also involved in blood clotting. In fact, the guy who actually first identified and synthesized serotonin worked with 10,000 liters of clotted cow blood to get enough to characterize it. He had a postdoctoral fellow, a research assistant, working in his lab named Betty Twarog. And she had an assay for serotonin she'd developed, and she used an edible mussel called a quahog. And it has a muscle in there that will contract in the presence of serotonin. So you can take this mussel, dissect it out from the quahog, tie a piece of thread to each end, hang it up in a glass organ bath that contains artificial cerebrospinal fluid, and hook a force displacement transducer on the end of it. You can measure how tightly it actually contracts. And put serotonin in there, and increasing concentrations, you can see the contraction increase and increase and increase. So she was assaying all kinds of animal tissues. And she went to Irwin Page. He was working in his lab at the Cleveland Clinic, and she says, I want to analyze cow brain, see if there's any serotonin in cow brain. He said, there's no serotonin in the brain. Everybody knows serotonin's all in the gut. There's none in the brain. He didn't even want her to do the experiment, but she did anyway. And sure enough, there was serotonin in cow brain. Now, you don't have to be an organic chemist to look at the structure of serotonin and look at the structure of LSD and know there's a similarity. And by 1953, the word was out on LSD. Sandoz had been supplying it to therapists as producing a model psychosis in their patients. So they were supplying it to everybody willy-nilly that had any kind of degree to try it out and see what it was good for. So imagine 10 years later they find serotonin in the brain, and now this is the aha moment. All these people that thought that schizophrenia was related to bad parenting suddenly had to admit, serotonin's in the brain, LSD has these profound effects, maybe neurochemistry in the brain has something to do with behavior. Now today that we think, well, of course, it's preposterous, they didn't know that, but they didn't know it then. And if you look in the National Library of Medicine, 1953, all the references are about serotonin in the gut as a blood clotting factor and so forth. And then 1954, 1955, 1956, you start seeing people analyzing the brains of different species of mammals to see where it is, what it's doing, and suddenly it skyrockets. So all the drugs today that are used for treating serotonergic problems, like depression, the SSRIs, drugs for migraine, all were developed because of the awareness of this serotonin being in the brain. So a lot of those medications would not have been developed, maybe we'd be better off without some of them, but wouldn't have been developed had LSD not been discovered. So LSD has its own importance as far as being a potent psychoactive, but it was also the catalyst that made people realize that brain chemistry had something to do with behavior. How do mescaline, phenethylamines, tryptamines, psilocybin, DMT, or LSD, how do they actually work? What they target is a serotonin receptor in the brain called the serotonin 2A receptor. And you see, the phenethylamines basically only hit 2A and 2C receptors. And we know 2C receptors are not really involved. There have been a lot of pharmacology studies to show that. Tryptamines activate 2A, 2C, and also 1A receptors. So we think serotonin 1A receptors are involved in the actions of some tryptamines. LSD is pharmacologically a very promiscuous drug. It not only hits 2A and 2C receptors, 1A, and hits all these other receptors. And that's one of the curious things. We don't know what role any of those other receptor interactions actually play because LSD is a very complex molecule. But the 2A receptor is the one that we're focusing on. Most people are focusing on research. So the question is that receptor expressed someplace that would make sense that the things that we see with LSD would be involved with the 2A receptor? Well, if you look at the distribution, this is a human brain that's been sliced across. And what they've done is incubate it with ketanserin, which is a drug molecule. It's radioactive. It has a tritium atom in it. And they put it on a film, and they look at where the radioactivity concentrates. And you see these red layers. This is where the highest concentration is. It's in the outer layers of the cerebral cortex, all throughout the cortex. So when I was a graduate student, I started working on psychedelics in 1969 as a graduate student. So I've been doing it longer than I think anybody. Sasha Shulgin did it longer than me, but he's dead. He's dead now, so I inherited that title, I guess. How do we test them? And I would go into my chemistry classes, and I'd have a graduate student on the bus, and he'd say, yeah, what are you working on? I'd say, oh, psychedelics, mescaline analogs. You need any volunteers? And that was a pretty constant theme. You need any volunteers? I said, no, can't do it. It's illegal. So how did we actually do some of the research we did? And I actually was lucky because I had grants from the National Institute on Drug Abuse for 28 years to study psychedelics. I was one of the very few people in the United States that was funded by the government to study psychedelics. And of course, they didn't want me to find better psychedelics. What they wanted me was to find things were wrong with them, to find out how we could stop people from using and so forth. But nevertheless, I had a big group and we had fun studying these. So how did we test them? So normally, if you want to find out what a receptor does, you develop a test that involves a modulation of that receptor by some drug. So for example, in the heart, you have what are called beta-1 adrenergic receptors. So if you don't know what they do, you develop a drug that stimulates those beta-1 adrenergic receptors, and you find out if you put that drug into a human, it stimulates the heart and makes the rate go faster. So if you want to study consciousness, doesn't it make sense you would use drugs that would modify consciousness? Try to figure out what consciousness was by using these drugs. So we learn about physiology by using drugs that perturb these systems. So the hypothesis was to study consciousness, we can use agents that perturb consciousness. That is psychedelics, which are some of the most potent psychoactive drugs known. Take these, I want to see what they do to you. And if any of you, can I see a show of hands? How many people have a copy of PCAL? Yeah, a few people. PCAL and TCAL are books. PCAL stands for phenethylamines I have known and loved. TCAL stands for tryptamines I have known and loved. And PCAL is a story, it's autobiographical, of Sasha Shogun and his wife in the first part of the book. The second part of the book are the chemical syntheses of all the drugs that they took. So Sasha would take a drug, and he would say, this is an interesting molecule. And he would go to his wife, Anne, and say, Anne, you should try this. So Anne would take it, and she'd either get excited or not. And she'd say, yeah, Sasha, that's great. And he had a group of friends who lived in the Bay Area, San Francisco, of course, where else would you find them? And so he would go to his friends and say, okay, Anne and I have taken this, and we think it's really interesting, and we're going to have you all try it. So they get together in one of their friends' homes every so often, and he'd weigh out a sample, and everybody would take them, and they would record the effect. So those records are in PCAL, and also TCAL for tryptamines. So if you ever get a copy of PCAL or TCAL, you can read about Sasha Shogun and Anne's autobiography. And then the second half of the book is all these drugs, how he made them, and what the responses were, what people said. I couldn't do that. Unfortunately, we had to use animals. And our animals never have spiritual experience. So it's a little bit of a problem. I was in a panel, an MDMA panel at Stanford University years ago, and we presented information about MDMA, and I talked about some of my synthetic work and animal studies. And in the Q&A afterward, a fellow asked me, or he asked the question of the panel, so you've talked about all this other stuff, synthesis and everything, but you haven't talked about the spiritual aspects of MDMA. And Sasha looked at me, and I looked at the other panelists, and we didn't know what to say. So I leaned up to the microphone, and I said, well, my rats are all atheists. We use something called two-lever drug discrimination. And this is a box. These two sides have been cut away. There's a light in here that lights up. And this is a food pellet delivery trough. There's a little solenoid that can kick a food pellet down. It's a 40 milligram sucrose tablet. So it's literally a little rat candy. And these levers operate micro switches. So the rat can push on the right or left lever, and it'll activate a micro switch. Now, rats normally don't press levers in the wild. So first you teach a rat to press a lever. You just turn one of these on, you put him in a cage with one lever, and every time he presses a lever, he gets a food pellet. And it makes a noise when the solenoid kicks it in. Pretty soon he associates the sound with the rat candy, and he learns if I press this, I'm going to get a rat candy. After he learns to press, then you differentially reinforce. So we associate one of these levers with the administration of something like LSD. And the other one we associate with placebo, or just saline, inert. So we put the rat in here on day one. We turn this lever on, and we give the rat LSD. Well, he knows if he presses a lever, he'll get a food pellet reward, but he doesn't know which of these levers does what. So he pushes back and forth and pushes back, and pretty soon he finds out that only if he presses this one does he get a food pellet. And then we put him in on day 2, and we give him a placebo injection. And we turn this lever on. And then he finds out, well, this lever doesn't do anything, this one does. We alternate drug versus saline back and forth. And after 2 to 3 months of training, the rat very reliably will learn. If you give him LSD, he'll go in and press this lever. If you don't give him LSD, he'll go in and press this lever. And this is a robust assay. You can use very low doses that don't produce any other behavior in the rat. And the rat will press 2,000 to 3,000 times in 15 minutes. If you ever saw one of these rats inside, they just go nuts. In fact, we used to have them trained so if they press once, they get a food pellet. But those food pellets, like all scientific equipment, are really expensive. And we were running through a lot of rat candy. So we made them push 50 times to get one. And they still went nuts. They still pressed like crazy. So what we did are made analogs of LSD. So we took LSD and we modified the structure, and I'll show you some examples. And then we put a rat in, and if he pressed on the lever that we had associated with LSD, he basically was saying, I think you gave me LSD. That's the assay we used. And it's quantitative. We have large colonies of rats. We could look at how many rats responded at different doses, get dose response curves. And then from human studies, we knew that had been published that this generally correlated with human activity. But what I'm saying is, this is a pale reflection of what happens when a human takes LSD. Well, I think he gave me LSD. What does that mean? But it allowed us to do research for years. We published many, many papers developing analogs of LSD and tryptamines and phenethylamines using this assay. And you can use any psychoactive drug. We use mescaline, MDMA, amphetamine, methamphetamine, LSD, any drug that has a central effect in humans. You can train these rats, and they're very sensitive. It takes a very low dose. And the other thing that's powerful about this assay is if you train this rat on LSD and you give him MDMA, he won't press the LSD lever, he'll press the placebo lever. The drug you give him has to be like the drug you trained him on for him to press that lever. So we were pretty sure that we were getting reasonable results. Now that's an expensive assay. The rats are expensive. When we started using this assay, rats were $2 or $3 a piece, and now they're like $50 a piece. So the alternative is a mouse assay, which is what people use now, the mouse head twitch. If you give a mouse a psychedelic, he'll shake his head back and forth. You have to video these mice, and somebody has to score it. Well, a colleague of mine, Adam Halberstadt, who's at UC San Diego, found out that he could open up the skull, the skin of the skull, and plant a powerful magnet under the skull, suture back up, put the mouse inside of a tube that had a big coil around it, and when the mouse would rotate his head, it would generate a current in the coil, and you could see it here. So the mouse head twitch is what people use because you don't have to train the mice. You just have a bunch of mice that have these neodymium magnets implanted and glued onto the skull. That assay, here's LSD showing the number of twitches with time and dose. And here's a DOI, which is a phenethylamine. And you see with doses, a low dose, you get nothing with 0. And then as you increase the doses, all these are head twitches. So you can actually do a quantitative measurement. So this is the kind of problem you have working in this field when you can't do clinical studies with humans. But it got me grants funded, so. LSD is a weird molecule because if you change this diethylamide to almost anything else, its potency drops off by about 10-fold or more. So these are 2 ethyl groups. So if you change one of these into a methyl ethyl, its activity drops off. 2 methyl groups, activity drops off. Methyl propyl, which has the same number of carbons, a single carbon here and 3 over here instead of 2, activity drops off. Anything you do decreases activity. So years ago, we started thinking, whatever this receptor is, if it's a 2A receptor, there must be a specific place where it binds. It really likes this diethylamide for some reason. And then we made a compound here that's got 4 carbons, but they're not diethyls, and we could introduce stereochemistry. So this is the R isomer, where the hydrogen is pointing out toward you. And this is the S isomer where the hydrogen is back to the screen. That's the only difference, the same molecular weight, same lipid solubility, etc., and about a 4-fold difference in potency. So we knew there was some kind of a stereochemical play going on there. If you tied the two ethyl groups together at the end into this 5-membered ring, activity drops way off. And this is known from human studies. Any change really knocked the activity of these lysergic acid amides down. Our only explanation seemed to be there must be something in the receptor that really likes a diethyl group. So we thought of a way to try to test that idea. We made some analogs where these two ethyl groups are floppy. They can swing around. So we said, let's lock them so they can't swing around. Let's lock them so they can be in specific orientations. So we could do that by taking this carbon and attaching it to this carbon with another carbon. So now you get a dimethyl azetidine. This is called an azetidine, and these are two methyl groups. And you can see these dots showing what would be the diethyl group there. And the beauty of that is, with the dimethyl azetidine, you can have the two methyl groups on the same side of that ring, or you can have them on different sides where you have the RR or where you have the SS. So that locks the diethyl groups. And this is shown over here. Here's LSD. Here's this one conformation where we have the front ethyl group pointed down to the 7 o'clock position and the back one up. Or you can flip it and you have the front one up and the back one down, which you can't see. And here's the SS. You can see the similarity when we have an SS versus an RR. If we look at the top view, here's LSD with these two different orientations of the ethyl groups. And so in one case, down and to the left, up and to the right, down and to the right, up and to the left. And here's the SS and the RR. This LSD, by the way, a lot of the compounds we made became research chemicals and designer drugs. This is sold on the black market as LSD now. Anyway, we made these two, and the chemistry was pretty difficult. We tested them in drug discrimination in rats that had been trained to discriminate LSD, and the SS was the one that was active. So that showed us there is a stereochemical preference, and those two ethyl groups bind in a specific orientation. So here's just indication. Here's LSD, and here's the azetidides. Here's the drug discrimination, ED50, measure of potency, LSD 45. The SS, 25, about the same potency. The cis drops off, and the RR drops way off. So in this in vitro, this is an in vitro assay, an enzymatic in cells, and we see the same thing. So then we had a model we had built of the serotonin 2A receptor. We had a model of the serotonin 2A receptor, and we used a computer to dock the LSD molecule in there. And what we noticed was, I don't know if you know anything about G protein coupled receptors, but there's 7 alpha helical pieces of protein that are bound in the membrane, and they connect on the tops and the bottom as it threads through the membrane, from the outside to the inside to the outside, etc. And there's a piece of the receptor that connects the top of helix 4 to the top of helix 5. And this leucine 229 was in that piece, and it was laying right near the diethyl group. So we thought maybe the leucine 229 is involved in directing those two diethyl groups. So then in 2017, in the lab where I worked, we published the crystal structure of LSD bound to the serotonin 2B receptor. The serotonin 2B receptor is very similar to the 2A receptor. Here's LSD, the red molecule. Here's leucine 229, and here's that extracellular loop 2 that folds over the LSD molecule. And this is LSD in the crystal structure in a representation. And if we superimpose that SS azetidide, it exactly superimposes. And if it was the RR, this blue group would be up here, and this would be here. So we predicted in 2002 the orientation of the diethyl groups in LSD that we verified in 2017 by having the actual crystal structure. So LSD is a very long-acting compound, much longer than psilocybin. And so we had a colleague who did computational studies, molecular dynamics, and looked at this loop. This is extracellular loop 2 that closes over the LSD molecule. And this is the percent of time that is open when there's nothing in there, there's no LSD, or when there's LSD bound in the wild type. And you see it doesn't open very much. It's only 2% to 1%. But if we mutated that leucine, in the 2B receptor, it was 209 instead of 229. If we mutated that to an alanine, which is a much smaller residue, and then did the same studies, you see it's opened quite a bit. Suffice it to say, this is LSD in purple, and this is the SS in green. And you see they superimpose in this case. And also, when you look at the ability of this mutation to affect, there's another signal that's produced called arrestin. If you look at the wild type, it takes about 8 hours for LSD to come out of the receptor. But if you mutate that leucine to an alanine, LSD comes out in about 2 hours. So through a series of experiments, what we showed was LSD has a lot of trouble getting into the receptor. And once it gets in, it has a lot of trouble getting back out. So that's the basis, part of the basis for the fact that it's so long acting. It takes a while to kick in, and then it takes a long time to get back out of the receptor. And it is time dependent. There's something called a beta-arrestin recruitment. And if you look at this 5 minutes, 30, 60, 120, this is increasing potency going to the left and increasing effect up in this direction. The longer LSD stays in the receptor, the more of this particular signal it generates. So LSD is a very complex molecule. It does all kinds of things to the receptor, including getting trapped in there. And the longer it's trapped in there, the more it changes these signals. So in 1993, I was really annoyed by the fact that nobody was doing clinical studies of psychedelics. So I got together, I called some colleagues that were interested in this field, and I said, let's start an institute, a not-for-profit institute, go out to philanthropists, get money to fund clinical studies of psychedelics. And nobody poo-pooed me because I was a full professor at Purdue University. And they thought, Nichols wants to study psychedelics? Like, he's at Purdue, he's got tenure, he's not going to ruin his reputation by doing something stupid. And the other people I got were also full professors, they were psychiatrists, fellow at UC San Diego, neuroscientists. So we started this. It's funded more than $9 million to support research. Anything you've seen with psilocybin, almost everything you've seen has been studies that we have funded and supported through the Hefter Research Institute. And years ago, a woman asked me, she said, what do you see in the future for this work you've been promoting? I said, someday, long after I'm dead, there'll be places where you can go and your doctor will tell you he's going to send you down to see a psychiatrist/shaman. You're going to be having a midlife crisis, and you'll go down there and he'll give you a psychedelic session and you'll get a new perspective on your life. And she said, you'll be dead, you think? I said, well, you know, at least if the vector is going the right direction, that'll be a good thing. Actually, I thought I would be dead before any of this happened. So just to show you some of the things, the first one is the first pilot study of psilocybin treatment in patients with advanced stage cancer, published by Dr. Charlie Grobe in 2011, a small study of N of 12. And then we had two big studies, psilocybin in treatment of anxiety and depression from a life-threatening cancer diagnosis, Roland Griffiths at Johns Hopkins and Stephen Ross at New York University. Those are both published in 2016. They got a lot of press coverage because a single treatment under the conditions of this therapy alleviated their depression remarkably for months and months and months. So this is the first indication in a large study that psilocybin in conjunction with therapy could treat effectively depression. Now there's been a study in Imperial College with just treatment-resistant depression where they've shown the same thing. Another small study that hasn't been published in just depressives, not end of life, we see the same thing. A single 1 or 2 treatments gives long-lasting relief of depression for months or possibly years. Matt Johnson did a study of psilocybin in cigarette addiction. Patients who had made 4 attempts to stop smoking had been unable to do it. He treated 15 patients and showed that 60% of them stopped smoking. This is remarkable. He gave them more than 1 treatment, but he had a whole protocol. He's expanded that study now, and it's at Johns Hopkins. We're doing brain imaging before and after. Psilocybin in treatment of alcoholism or alcohol use disorder. Dr. Michael Bogenschutz at University of New Mexico did a small pilot study, N of 10. He's now expanded that study. He's enrolling 100 patients, our subjects, our participants. When I spoke to him a month or two ago, he had already treated 80, seeing the same kind of results. Treatment for alcoholism. Studies of the effects of psychedelics on gene expression and anti-inflammatory effects. My son, unfortunately, is following in my footsteps. He's at LSU, and he discovered accidentally that psychedelics are extremely powerful anti-inflammatories. In a mouse model of asthma, he can both prevent asthma and he can reverse asthma. He's looking at all kinds of inflammatory conditions. Atherosclerosis, it'll reverse atherosclerosis. So he's got patents on that, and there's a company now that's trying to commercialize that. So he's looking at also the gene expression changes. What do they actually do to the genetic expression? Let's see, Franz Vollenweider, we have a clinic that Hefter supported in Zurich. He's published 30 or 40 papers looking at the mechanistic basis for emotional and mood regulation with psychedelics. We also have Peter Hendricks, who came in late today. He'll be talking in a panel at least, and maybe, I guess he's given a couple talks, treatment of hardcore cocaine addicts with psilocybin associated with therapy. Also a study on long-term meditators from Johns Hopkins, showing the effects of psilocybin therapy on long-term meditators. So this is like really opening up. The FDA has given breakthrough therapy status to psilocybin for treatment-resistant depression. You probably all know MDMA has gotten breakthrough therapy status for PTSD. I made all the MDMA that MAPS used for all those studies. Being a chemist has some advantages. So I hesitate to call this the philosopher's stone or the golden or magic bullet, but it's really amazing that these things do all this. And the tragedy is for 50 years, politics kept any of this from happening. And we still can't get these studies funded by any of the national institutes. National Institute of Mental Health, National Institutes of Health, they won't fund these yet. But we hope they will when some of these other studies are published. So if psychedelics can effectively treat all these things, what's going on? Pharmacotherapy today is empirically based. Someone's depressed, you'll go in, your primary care physician will put you on an SSRI. He'll try it for 3 or 4 weeks. If you can't tolerate it, doesn't work, he'll put you on a different SSRI. If that doesn't work or you can't tolerate it, he'll put you on SNRI. If that doesn't work, he'll put you on bupropion. He'll try all these things. It's empirically based. It's trial and error. Schizophrenia. We don't know what the basis for schizophrenia is. Well, dopamine receptor blockade seems to improve symptoms, but how is that involved? We don't understand the basis for any of this. All the drug companies have got out of development of CNS drugs. They're all making biologics, things for arthritis and all kinds of things, diabetes. They're not doing CNS drugs anymore because they don't know what the targets are. They don't understand what causes it. So there's going to be a change in the paradigm in the way we approach these with psychedelics. So traditional notions of receptor-based pharmacotherapy are not explanatory. Something else is going on. And the way the brain operates, let me try to use an analogy. If you have a state or a small country that has a lot of little cities, big cities, small cities, villages, each one of those little villages would be self-contained and integrated within that village, and maybe they don't send communications out to the other villages or cities very often. Your brain is like that. You have anatomically defined areas all over your brain. They're constantly going about the business of whatever that particular brain area is supposed to do. Those areas or nodes, communication nodes, are integrated within the node. They're all doing the business they're supposed to be doing, and they don't reach out and talk to other parts very often. So that would be represented like this. Now, this is not anatomically correlated, but each one of these little round colored things is a communication node or hub. And the energy involved in that hub is represented by the size of those circles. And you see there isn't much communication going on between these different hubs. We now know from imaging studies of all kinds of psychopathologies that when someone has depression, obsessive compulsive disorder, or whatever, you have a failure of these hubs. Things start communicating that are not supposed to be communicating. Well, when you give a psychedelic, like psilocybin, all the integration within those hubs breaks down, and you generate a global communication. All these parts start communicating with each other. And it's like a reset. If you've ever had a PC, you had to go do the Control-Alt-Delete. It's something like that. But basically, we need to think about the brain in terms of brain dynamics instead of receptor-based function. So psychiatry is going to undergo this revolution. It's starting now. So psychedelics have helped to spur that by giving us insight into what's going on when a psychedelic hits the brain. And then when it leaves the brain, presumably these dysfunctional hubs fall back into their genetically determined patterns. But we've known about some of these things since about the late 1900s. And we know that glutamate is a powerful transmitter that's involved in a lot of regulation in the brain. And one of the things that occurs is an area called the colostrum, which is the most highly connected area in the brain. The colostrum connects all these areas, and the colostrum, when a psychedelic hits a colostrum, it starts generating impulses. And that goes out to the whole brain and will help to change the connectivity. We don't understand exactly what happens there. That's the big question for psychiatry, but that's what psychedelics are leading us to, understanding the basic underlying mechanism for how the brain actually works, coupled with brain imaging techniques. We do know from studies with ketamine some of the receptors that are involved in depression and how that's reversed by ketamine. And we see things, generation of new synapses and axons in response to psychedelics. So people are starting to look at the molecular and cellular events that are involved in producing these treatment effects. So I'll leave you with this slide. I hope at least most of that was intelligible. And of course, I didn't do all this work. I had 45 graduate students and 31 postdocs and visiting scholars. And NIDA funded most of the work that you saw here. And this is just a tiny bit of what we did. Obviously, it would take a book to cover everything that we did. So thank you for your attention.